And now we're going to look at the endoplasmic reticulum. The endoplasmic reticulum has a lot of membrane in the cell, and all that membrane must be doing something. The vesicles that travel from the ER to the Golgi are COPII vesicles. That is, their coat, the protein that helps them form and pinch off from the ER, is made of the protein COP2. And the process by which this occurs is similar to that of the clathrin-coated vesicles, in that receptors in the ER contain an exit ER address. These receptors then bind cargo proteins that also have an exit ER address, and these cargo receptor complexes then recruit adapter proteins that cause the COP2 proteins to be recruited and for the vesicle to form. As the COP2 vesicle pinches off and leaves the ER, the COP2 coat depolymerizes and the vesicles fuse to form the vesicular tubular clusters, which will then become part of the Golgi. The COP2 proteins, which are in the cytosol, will go back to the ER to be reused, and any proteins that are present in the vesicular tubular clusters that contains an I should be in the ER address will be sorted and returned to the ER. The vesicular tubular clusters are then moved to the cis face of the Golgi using traction motor system. What I've just described highlights a key point. Trafficking is not all one way. That is, it is not all from the ER to the Golgi to the plasma membrane, so-called anterograde trafficking. There is also return trafficking, retrograde trafficking, to return proteins and membranes to their start positions. If there wasn't, then all membrane-bound proteins and membrane would accumulate in the plasma membrane, and the plasma membrane would get bigger and bigger, and the ER and Golgi would get smaller and smaller as they lost proteins and membrane. Finally, there are three other things you should remember about the ER. One, it is the site of disulphide bond formation in proteins, and these bonds will form as the protein is co-translationally imported into the ER. Two, protein glycosylation is started in the ER and finished off in the Golgi. And three, the ER carries out quality control on proteins and will mark proteins for destruction that are not correctly folded, and also will often incorrectly mark some correctly folded proteins for destruction.